Hello my dear students, welcome back to Biology at Priyajit. Today we are starting one of the most fundamental and fascinating processes of developmental biology, that is cleavage. Have you ever wondered how does a single cell, the zygote, transform into a complex organism? So the answer begins with the cleavage. So what is cleavage? Fertilization gives rise to a zygote and immediately after that begins a rapid series of cell divisions called cleavage. Cleavage is the repeated mitotic divisions without growth. The zygote divides into many smaller cells called blastomeres. These cells remain tightly packed. Eventually they form a hollow structure called the blastula. Key terms: blastocele, there is a cavity found inside the blastula, and blastoderm is the outer cell layer. So remember, cleavage prepares the embryo for the next stage, that is gastrulation. So you have to know the role of yolk in cleavage. We know that the topic now comes a very important concept for our exam purpose. Cleavage basically depends heavily on yolk content. Yolk is basically the stored food material, also called as deutoplasm. So active cytoplasm divides faster than yolk rich region. So what is the key effects of yolk? When more yolk, less active cytoplasm, the nucleus shifts towards animal pole and it leads to unequal cleavage. And when yolk shows down division, yolk rich cells divide slowly and it results large blastomeres at vegetal pole. The important rule: more yolk, so the process will be slower cleavage will be occur. So the cleavage is basically the rapid cell division that leads to a multicellular embryo. Though so you now we have to know the planes of cleavage. Cleavage is not random, it follows specific planes. These are the various planes. First one is the meridional plane. The cleavage furrow passes from the animal pole to the vegetal pole. Vegetal pole is the pole of the dividing cell that contains the yolk material. So meridional plane is the cleavage furrow, passes from the animal pole to the vegetal pole through the center or the median axis of the egg. It divides the egg into two equal halves. Example: first cleavage in frog and chick. So this is the animal pole or AP, this is the meridional plane, and this is the vegetal plane. So it divides the egg into two halves. In case of frog and chick, this is the first cleavage stage. Vertical plane, the cleavage furrows also runs from animal pole to vegetal pole, and this this cleavage plane is smaller than the meridional plane. It does not pass through the central axis, it goes through the off center, so the resulting cells may be unequal in size. So this is the third cleavage in chick. Equatorial plane, the cleavage furrow runs at right angles to the animal vegetal axis. It passes exactly through the middle of the egg. It divides the egg into upper and lower equal halves. So example: third cleavage in sea urchin. Longitudinal or transverse or horizontal plane. Here the cleavage furrow runs parallel to the equator or to the horizontal direction, but it is slightly above or below the equator. It produces unequal cells. Upper cells or micromere are smaller, but the lower cells or macromeres are larger due to more yolk content. So examples include amphibian eggs. Types of cleavage based on fate of cells: determinate or mosaic cleavage, cell fate fixed earlier. Each blastomere have the specific structure. It is common in protostomes. Indeterminate or regulative cleavage, when cells retain full potential, one cell is complete organism possible. It is seen in deuterostomes. Based on extent of cleavage, it can be holoblastic cleavage or complete cleavage, and meroblastic cleavage or incomplete cleavage. Holoblastic cleavage is the entire egg divides. It is found in eggs with less yolk. The types of holoblastic cleavage are the radial cleavage, which is found in echinoderms or amphioxus. Spiral cleavage is found in annelids, mollusks, flatworms, etc. Bilateral cleavage is found in tunicates, and rotational cleavage is found in nematodes and mammals. So examples: human shows also rotational cleavage. Meroblastic or incomplete cleavage, only part of egg divides due to presence of yolk. It is incomplete cleavage, and due to heavy yolk content this happens. Types: it can be discoidal cleavage, blastodisc forms on the top of the yolk. A disc-like structure is found in birds and reptiles. Superficial cleavage, nuclei divide first, then cytoplasm. So it is found in insects like Drosophila. Special cleavage patterns. So the types: radial cleavage, it is symmetric. Spiral cleavage is oblique divisions. Rotational cleavage found in mammals, and bilateral cleavage is the produces the mirror image hubs. Rules of cleavage, very important for theory questions. Sachs rule: cleavage planes intersect at right angles. Hartwig's rule: spindle aligns along longest axis. Balfour's rule: rate of cleavage should be inversely proportional to the 1 by Yolk amount. Fluger's rule: cleavage follows path of least resistance. So final concept: why cleavage matters. Cleavage is not just division, it is organization. It produces enough cells, establishes body plan, and it prepares for gastrulation. So my dear students, we build the foundation of embryonic development. If you understand this clearly, then trust me, gastrulation will become much more easier for you. So like, share and subscribe to Biology at Priyajit. Comment cleavage clear. If this helped you, see you in the next class. Thank you.